It's a pleasure to participate in this scientific session by ALMA. I have been involved in several such initiatives with the International Federation of Fertility Societies, and Sergio and my colleagues in the ALMER organization. We're very proud of it, and we're very happy to participate again. I've actually been given a fairly difficult topic, the genetic causes of premature ovarian failure, which is synonymous with the term premature ovarian insufficiency. And that's the reason. The reason is that it's changed over the years. At one time, we thought we were fairly familiar with the causes of ovarian failure, namely X chromosomal abnormalities. And certainly over the last 10 years, we've learned that there are other explanations that are much more prevalent. The disclosures I have are listed on these slides, namely being a member of the clinical expert panel for Illumina, which, as I think many of you know, makes most of the DNA sequences in the world. What we will do here is discuss the causes genetically of ovarian failure. I think we're all familiar with the diagnosis and the approaches here. Elevated FSH and elevate and LH make the detection and the definition quite easily over a given level. Historically, we've thought of these women having short stature, characteristic somatic anomalies that are common in Turner syndrome 40X, 45X. There are also variants that occur if you have lost only a portion of the X chromosome. When we see this in a woman with the normal karyotype, we do not expect to see short stature, and we do not expect to see somatic anomalies, although it may happen. A variant of the theme for complete ovarian failure occurs if menstruation begins, but it stops before the expected age of the menopause. And in particular, the definition is applied to women under the age of 40 whose menstruation has stopped. So here is the classical patient with Turner syndrome 45X. This frequently occurs in the context of more than one cell line mosaicism. It also occurs in the context of a portion of the X chromosome being missing. So these eras point where there could be a break point with loss of the chromosomal material on the X chromosome distal to that. So for example, if you had a loss right here with this break toward the tip of the short arm of the X chromosome, all of the X would be present down toward the bottom here, but not the areas here. So for many years, we thought this was the best approach to identify specific genetic regions much shorter than a complete chromosome that contain pivotal loci. So once we did that, we tried to break this down into primary amenorrhea, secondary amenorrhea, and you can see that the stratification was simply not that nice, and that's probably because mosaicism existed. Occasionally, we did have a region that we decided was important, and we could identify a gene related to that. And you can see this one was a gene called bone morphogenetic protein. It's not really important to remember that, except the concept was valid, but it was not very efficient in identifying that. So very quickly, we learned that the genes that were controlling ovarian development were located not exclusively on the X chromosome, with or without mosaicism, but they had to be elsewhere, and that elsewhere related to the autosomes, chromosomes 1 through 22. The sequencing strategies, therefore, became necessary. We had to be able to sequence the DNA, identify exactly which portions of the DNA in the complete genome were missing, and therefore we could identify a gene that was important and moving to the future perhaps could be replaced in women who had ovarian failure, premature or otherwise. But this was slow going, and actually until about 15 years ago, we had very few situations in which we were convinced that there was a gene causing premature ovarian failure that was not on the X chromosome. So one that we did know about was a phenomenon that occurred in Finland. In Finland, it's very common for women with 46XX normal karyotypes to have their premature ovarian failure due to an autosomal recessive gene. We knew this because of the statistics on the slide. There were many aggregates, particularly in a specific region of Finland. The consanguinity rate was very high, and the ratio of affected females in a family was about the expected Mendelian ratio of 25%. So this actually set the stage for Dr. Edamaka in Helsinki to be the first to show and to discover a proven autosomal gene that was responsible for premature ovarian failure. And this gene turned out to be in the receptor that responded to the pituitary secretion of FSH. So all gonadotropins and all hormones in general have to have a receptor so that as they circulate in the body, they reach the end organ in question. And in that particular situation, they produce the products that are necessary. So in this particular situation, in the extracellular component of this G protein receptor, the FSH receptor, there were two mutations, missense mutations, that resulted in a valine rather than an alanine here. And so this produced the ovarian failure that was noticed clinically. It turned out, however, that this was very uncommon. It was common in Finland, but almost nowhere else. And actually, it's a reportable case to find this receptor defect in a country other than Finland. So this demonstrated the proof of principle, but it certainly was not a common situation that would have existed. So other strategies need to occur. And fortunately, they coincided with the fact that the human genome had been sequenced and was applicable. And it turned out that it was not quite as expensive as we had imagined in the beginning. So that at least by the latter part of the first decade in this century, it was possible to take a region of the chromosomes, sequence that region, and determine whether or not the DNA nucleotides that were in that area were what we expected or whether they were abnormal. And if they were abnormal, were they consistently observed in more than one individual, indicating that this could actually be the molecular explanation for this particular condition or so. So one of the areas that we could look at right here would be that if we did not find an explanation for premature ovarian failure or any ovarian failure by looking at hormone regions such as receptors, the hormones themselves, and the enzymes involved in hormone biosynthesis, all of these were logical, but we didn't find them very much. So the next step was to turn to look for these causative genes along the pathway of embryogenesis. And we would take advantage, as I've just mentioned, of the fact that DNA sequencing technologies exist and they could be targeted. So the logical thing to do is to go back and look at our embryology. All of us know that primordial germ cells originate in the epithelium of the yolk sac. We don't think about it very much, and it's not a clinically relevant fact to relate to patients. But nonetheless, we do recall that they migrate down toward the different gonads, and the primordial cells reach the gonadal ridge in a particular area, and an entire series of cascade of sequences occur to produce what we know clinically to be primary and secondary oocytes, which are utilized in obviously in life and in IVF in particular. So a few facts just to mention before we leave the embryology and talk about the clinical implications. The indifferent gonad develops into an ovary around embryological days 50 to 55, and the equivalent of the sex reversal Y gene, to which we talk about a lot, is called FOXL2, forkhead Hovia box. So at 20 weeks, it's important to realize that the fetal ovaries contain about 7 million germ cells. The vast majority of these undergo atresia, as indeed most oocytes undergo atresia in any menstrual cycle. And there are only about 400 that are actually ovulated in the life. The only real difference between 46XX normal women ovaries and 45X Turner syndrome ovaries is that the attrition is much more rapid in 45X than in 46XX. And it turns out that there are many genes on the X and many genes on autosomes that are necessary for this ovarian development. So this is a very complicated slide that shows the various genes that are involved in taking the indifferent gonad and developing that into an ovary. We're not going to go into that except to say that there are some of these that are important and they show up in patients who have a mutation. FOXL2 is one that is definitely worth noting or so. So there are some genes in this pathway that have dual roles. They are essential for ovarian development, these three that are listed on this particular side. But since they are so early in embryogenesis, they have a dual role assuring that the proper somatic tissue that's necessary in which the oogenesis is proceeding is developed. And when you have a dual role like that, a mutation of a given gene can have a disparate number of different features. So notice here, skeletal anomalies, or urogenital tract anomalies, and uterine anomalies. All of these are necessary in this if there's a disturbance of this particular gene. The one gene in this area that is worth clinicians remembering is FOXL2. That is the gene that must exist for ovarian development to occur. And we see it in clinical context. This young woman has somatic features. She has blepharophimosis. She has ptosis of her eyes. She has epicanthal folds. And this is an autosomal dominant syndrome that's recognized. It can also be recognized as an isolated explanation for premature ovarian failure. So the fact that there is a gene that functions with the resulting in somatic tissue as well as strictly the ovarian tissue is worth remembering clinically. So the issue here is exemplified by the name forkhead transcription factors. Transcription factors means that that's necessary to produce the DNA that's necessary. And in this case, that was not the case. So the gene is expressed in eyelids as well as oocytes. Exactly why, we really don't know. Let's move on to something that's more familiar. So here is a schematic diagram that shows the passage once you begin with the primordial germ cell and you develop into a secondary follicles. So clinically, we are much more familiar with primary and secondary follicles than the primordial germ cells, but they have this pathway, and we won't talk about it in details, except it forms a cyst as the germ cells are collected, eventually a primordial follicle, and then a primary follicle. Those genes that are noticed in yellow facilitate this process and are essential. And we've known this for a while based upon animal work, in which a given protein stops if there's a mutation beyond that level. So if you have a block here, you will have OCT4, nanos 3, but you will not have genes that are on the other end here, or at least in a particular pathway. So each of these has been analyzed and shown to be relevant to premature ovarian failure. And our own group, with a series of collaborators, have looked at patients and identified this in all of the genes that are shown in the red box right here. I particularly called your attention to this group here. We were together in about 2005 to 2008. Professor Ying Chen, shown in the left here, is the most active person in this field in terms of identifying particular genes, but all in this group whose names are displayed there have contributed in one way or the other. Dr. Chen presented to Samir within the last year and remains very active in IFFS and in lecturing around the world. Just to show you examples, no box is one of the first named actually by Dr. Rajkovic, who was the tallest in the picture before. Here's a study that we did. No box shows is the name newborn ovarian homeobox, NOBOX. It's fun to name these. This is a study we did at the time, 2007, in which U.S. Caucasian women were studied, and 2 out of 96 showed a missense mutation. It's shown in a box right here. We showed there was functional significance. We didn't see it in the controls. They were in a conserved region. We won't bother to go into it, but it's something we look for in determining cause and effect. And the particular mutation disrupts DNA binding. And so you would expect that it would have a deleterious effect. Indeed, it did have that. Here's another example of how you could go back to that schematic diagram that showed before, pick out a gene and a protein product that was necessary. Here, this one is called figula. I won't go into its derivation, but what happens here is that there is a deletion of AAC here. So once you have had that, you have skipped what you would expect. This is a wild type right here. AAC exists right here, and you go along the top here and you don't see the same thing. So once you have this particular deletion, you are missing a particular nucleotide, a particular amino acid, and you have a gene product that's there. It has a name right here. So the 3 deleted nucleotides, ACA, in this particular case, cause the loss of asparagine at position 140. So this is a symbol for asparagine there. It has been deleted. So this is how you prove that premature ovarian failure or complete ovarian failure in this particular situation is caused by this particular gene that has been mutated. It's a long time in coming in terms of how we have elucidated the explanations for ovarian failure, but the message is that we have the technology and we will continue to do that moving forward in the future. Now, what are the other explanations that can exist for premature ovarian failure? So overall, we can start by remembering that there are a huge number of oocytes, 7 million at 20 weeks, we have mentioned. These 7 million oocytes must be winnowed down to a few hundred, which are eventually ovulated for clinical significance. And the assurance that we have at least the optimal number at a point in time requires the transcription factors we have identified, we've noticed, and have identified for illustrative examples, the Knox-Knobox and Figlu, but there are others as we have seen here. The second is there is simply increased attrition compared to what's normal. We've talked about the 7 million winnowing down to about 400 to 500 in 46XX individuals. So some attrition is normal and required. On the other hand, in Turner syndrome, what happens is that the attrition is at a more rapid rate. It's the same phenomena of decreased number of oocytes, but it occurs much more rapidly. The other three areas are new, and we have not thought about these too much in terms of OBGYN, in terms of the explanation for lack of a full complement of oocytes. So, and this is the reason for this, is that we expected that the genes involved would be those familiar to us, such as hormones and FSH or FSH receptors. It turns out that there are a minority, only counting for about 10 to 15%. So the genes that we've discovered most often have been those that are necessary for chromosomal segregation, for DNA repair, and as one complement of a pleiotropic syndrome. A pleiotropic syndrome is one in which there are multiple copies and multiple organ systems that are involved as a result of a single mutation that may occur. Examples of this are familiar to us clinically. We know about Marfan syndrome as an example, tall, lanky, with the problems with the eye, problems with the joint. That's an example of a pleiotropic syndrome. It turns out that the genes that produce most disorders that are responsible for premature ovarian failure, in fact, represent DNA mutations and DNA repair. It's not a shock that this has occurred because, as I will mention in at least one slide, we've known that there are certain rare conditions in which a chromosomal breakage occurs, and they almost always will have ovarian failure. But we didn't think that the genes that we don't see as part of a syndrome were responsible for the same thing. So I'm not going to go over in great details what we now know to be about 50 different genes that are dealing with chromosomal segregation, DNA repair, or are part of a syndrome. I'm simply going to show you several slides that illustrate this component so that you can be quite familiar with that. So again, let's go back to think about it. Why are these genes, those controlling chromosomal segregation and DNA repair, why are they so necessary in ovarian development and assurance that this is appropriate? Well, there are millions of germ cells that have replicated, as we've mentioned several times. They're held in limbo for many decades while they're waiting to be used later in life, several different decades. So there will be requirements for DNA to be repaired as external damage to the germ cells will occur. They have to be repaired or otherwise they do not persist. And in addition, the atresia that occurs has got to be controlled. If there is no checkpoints and no process for this to occur, it is not going to produce the proper number of oogonia at the proper period of time. In addition, once fertilization does occur in IVF or in natural conception, very rapidly the embryo must be capable of vast numbers of cells over a rapid period of time. Think about going from the one cell zygote to the blastocyst, where there are more than 100 cells in just a short 6-day period or so. So I'm going to show you several slides, not going over any of these in details. Dr. Chen has written review articles on this. I have written review articles. All you need to do is look this up if you're interested, or look and do your own Google searches using the gene symbols that are shown. Hit Google and you will find that. So there are a series of disorders that are important to oversee the cell divisions. Obviously mitosis, obviously meiosis, which reduces the number of chromosomes from 46 to 23. These have to be perfectly timed in order for the process of cell division to occur, especially mitosis, which has to continue in some point in time, turn on and be very efficient. So a helicase is an important concept that's necessary for meiosis 1. We don't need to go into it, except a mutation produces ovarian failure. We can also look at what happens when the chromosomes need to come together and then undergo recombination to persist and stay together until they undergo cell division at the appropriate period of time, to make sure that there is a proper number of descendants for each cell division can occur. 46 in the case of mitosis, and of course 23 in the case of meiosis, gametogenesis. And also pivotal, as we alluded, is there has to be some signals that decide when you can proceed with cell division. It has to be at the right speed. So the concept is not to worry about the details here. It's the concepts to realize that as clinicians, ovarian failure is going to occur because of failure of the germ cells to be able to undergo division as they had expected to go. Here's another set of genes that are related to that. These are related to DNA repair. We know a lot about DNA repair genes because they produce cancer, gynecological cancers. We could list several. There's a role for BRCA1 and 2, our well-known breast cancer disease that can produce problems. So there are other situations as well. Many of these do have a relationship to cancer. For example, several of these along here, we can simply go down right here. Just think about meiotic recombinase is a mutation. You don't need to know anything about it, except it relates very much to the necessity of proper DNA repair. TP63 is another good example. So the point of these is not to memorize a name unless you are trying to get a PhD in this area. It's not going to be essential, but the fact that we need to think beyond hormones and beyond receptors for hormones as to what causes DNA repair defects that diminish the number of oogonia that's there. This is how we're going to help some of our patients in the future in terms of having effective oogonia. Here's a good example. I told you we suspected this for years, but all of these genes on this slide are associated with breakage of chromosomes. And if these children, because these are children in this case, have mutations in these autosomal recessive traits, they invariably show ovarian failure. We see the same thing in premature aging conditions such as progeria, Werner syndrome, Rothman-Thompson. All of these are associated with ovarian failure because of the reasons we mentioned. Finally, and this one is worth a few words of clinical value. In this particular situation, these syndromes have as one component premature ovarian failure. We've already mentioned blepharophimosis, ptosis epicanthus. There are several others that are down here. For example, progressive external ophthalmoplasia, PEO. This is a condition in which obviously you have the ophthalmological component to it, but you also have ovarian failure. Fragile X syndrome deserves a mention, just as breast cancer 1, BRCA1, in which case you also have POF or POI. So let me simply say a word about this very common condition, and then we will close our discussion. So X-linked recessive is due to an Fragile X syndrome is due to an X-linked recessive gene that's localized on the distal long arm of the X chromosome. We call it Fragile X because before the DNA days, we could recognize that it was a fragile site. It broke off because of a deficiency that could be put in certain culture medias that would exacerbate this. It is the most common disorder producing mental retardation in males, 46XY. In females, it's uncommon, but has a prevalence of about 1 in 4,000 to 1 in 8,000. And women can have borderline IQ, but they are far less severely affected than males, as you would expect in X-linked recessive inheritance. This is worth separating. The typical woman has 45 or less triplet repeats that are called cytosine, guanine, and guanine, CGG. It's not something that needs to be remembered. But the number of being less than 45, typically about 30 to 35, is worth remembering. Some women have more than that, but do not exceed 200. And this is called a premutation. The important fact of this premutation, as it is called, is that about 10 to 15% of these women will have premature ovarian failure. So it's actually, along with 45X and mosaicism, the most common explanation for premature ovarian failure. And during meiosis, the number of CGG repeats during female meiosis can increase, and it can exceed 200 in this diagram that you see right here. So this shows the actual Fragile X syndrome greater than 200, premutation less than 200, Fragile X clinical syndrome greater than 200 in this situation. So the summary of this is 10 to 15% of premutation carriers manifest ovarian failure. This accounts for about 1 to 5% of the sporadic cases and about 10 to 15% of familial cases. And again, we could talk at some length about the nuance, but the key point to remember is that you have normal ovarian function. It's just premature ovarian failure, increasing until you get to about 200, and then it stops and it slows off. So it increases up to a point, it stops, but if it's greater than 200 in that situation, you will have a full Fragile X syndrome in both males and females. One final slide. Early menopause, as well as premature ovarian insufficiency are probably the same condition. We imagined that could have been the case. We certainly knew there was an association, but now once that we have genes, we can see that in the same family with the same mutation, some women will have early menopause, that is, menstruation stops at, say, age 45, whereas others, often siblings, often offspring, will stop their menstruation under age 40 or so. And we can show that the same genetic variant in this situation is present in each family member in that case. So 2 final slides here showing what we've talked about. Premature ovarian failure and premature ovarian insufficiency. One is designated, is used to designate the number of genes. The other is what we're used to in clinical gynecology. So again, oocyte number is highest in utero. It undergoes attrition over a lifetime, and eventually only a few hundred are ovulated. This has to be a controlled process, just like ovarian stimulation. About 5% of premature ovarian failure is actually occurring in 45X women who actually do begin to menstruate. They're not all primary amenorrhea, and about 10% will have 45X, 46X mosaicism. Traditional endocrinologic explanations were uncommon except for the FSH receptor in Finland. There are a few genes that were perturbed in early differentiation and therefore affect both gonadal and ductal organs, and we've illustrated a few like that, but again, these are relatively uncommon. What's not uncommon is an explanation for premature ovarian failure is those that we see in these bullet points. At each step, as oogenesis goes from primordial germ cells to primordial follicles to primary and secondary follicles, transcription factors have to be intact. And we've listed and illustrated that. There are pleiotropic genes, which we've illustrated by Fragile X, that can, as one component, have premature ovarian failure or complete ovarian failure. But much more common as an explanation in 2021 is the fact that there are genes that produce errors in chromosome segregation, listed a few here, but also premature ovarian failure. There are genes that are characteristic of defects in DNA repair, and they also will do the same thing. Where will we go in the future? We don't expect anyone, including myself, to remember all these genes, but expect that we will be having available gene panels so that when our patients with premature ovarian insufficiency are developed and come to see us, we'll be ordering gene panels, and in that situation have a specific diagnosis. In the future, we would expect that there will be therapies that will be available replacing the gene product that's missing, because that's generally the pathway that we take for all genetic conditions. I hope this has been interesting in showing you this particular aspect of how genetic factors and how genetic technology can produce an explanation for conditions that we've puzzled about for a long time. Thank you very much.